production, and even their behaviors. And for example, because I'll be talking about heat for the entirety of my talk, rising temperatures can put pressures on species either to adapt to those warming climates or to migrate out or undergo range shifts in order to cope with those pressures. And so, as I've hinted, species need to respond to these climate change pressures in order to adapt and survive. So take this butterfly here. Prior to climate change, it was the favored phenotype of this dark morph. However, now that climate change is here and rapidly altering environments, it is now shifted so that this light morph butterfly is the favored phenotype in the environment. And really what I'm interested in is what are the mechanisms of response? So what is actually driving that change and allowing this butterfly or this population of butterflies to go from mostly dark morph butterflies to more light morph butterflies? And so there's two main categories of mechanisms that I'll talk about today. And the big one, first of all, is genetic changes. And then the other one that I'll spend most of my time on today is phenotypic plasticity. And so species with greater genetic diversity or greater phenotypic plasticity are more likely to survive and adapt to these rapidly changing environments induced by climate change. And selection pressures often favor individuals that can exhibit either flexible behaviors via phenotypic plasticity or have greater traits in response to new conditions. And so let's jump into genetic change, and in particular, how mutations can drive response to environments. So mutations can alter gene expression via permanent changes in the DNA itself. So take this code of melanin here. That particular DNA code codes for a dark morph coloration. However, if we expose populations over time and through multiple generations to elevated temperatures due to the rapidly changing environments due to climate change, eventually over time, as we all know, mutations will occur. So if you're very keen-eyed, you'll notice that there actually is a mutation in this sequence. And so the third nucleotide has switched from a G to an A. And we can say in this hypothetical example that correlates with a light morph butterfly. So that could mean that over time, this mutation, which is beneficial in response to climate change, may undergo selective sweeps, increasing in frequency, and may eventually possibly become fixed in the population because it's so advantageous given the new environment. But let's look, the mutations aren't the only way genetic changes can help adapt to environments. Obviously, mutations drive a lot of variation, but there's standing genetic variation that can be selected upon, and then integration as well. In complete opposite kind of way of responding, we have phenotypic plasticity. And so phenotypic plasticity does not require genetic adaptation at all or genetic change. And that's because phenotypic plasticity is the ability for an individual or one genotype to produce many different phenotypes when exposed to different environmental conditions. So let's just look at a real world example of this. So take these butterflies here. When they hatch or are born kind of in the wet season, they develop these really big eye spots that you can see here. Interestingly, the same exact species, if hatched in the dry season, develop these really, really small eye spots in a completely different phenotype. And the researchers here found that the difference in eye spots are due to the difference in predators that occur within the different seasons. And they used the seasonal cues of rainfall in order to kind of pick which phenotype is more advantageous for their given environment they're going to live in. And so what's really cool is phenotypic plasticity contributes to the phenotypic variation that's available for natural selection to occur upon, and therefore it can actually facilitate, hinder, or even preclude genetic adaptation, which is really cool. So it actually works within and with genetic change. And so the major molecular mechanism that drives phenotypic plasticity, and the one that I'll be spending a lot of time on today, is epigenetics. And epigenetics are cool because unlike genetic mutation, they actually can alter gene expression without altering DNA sequences. And there's a lot of different types that people study. So one big guy that you've probably heard of is histone modifications, but there's also non-coding RNAs, and the one that's near and dear to my heart that I'll be focusing on for the rest of the talk, DNA methylation. And because I'll be talking about it so much, I just wanted to go over how DNA methylation actually works. So take this hypothetical example again here. So we have the code for melanin, but we know that it's not going to undergo genetic mutation. A different mechanism is going to take place in order to change the phenotypes here. So once again, we're going to expose these butterflies to heat, but you'll note that instead of being within multiple generations, this actually can occur within a single individual's life. So one butterfly. So instead of thinking about it as like a systemic change of heat, think about it more as like a heat wave. It's going to occur within one individual's life. And so after exposing them to this heat, what these individuals can actually do is add a methyl group to the cytosines within its genome. And this can occur genome-wide, but in this particular case, it occurred within a code for melanin, and that addition of that methyl group actually changed the gene expression of this gene. And you'll see now the individual looks like a light morph butterfly. But what's really cool, and why I've chosen to really think about DNA methylation for my dissertation, is that if we take away that heat, what can happen is that individual is actually able to go through and undo the changes that it once did. So it removed that methyl group. And what that does is that it allows for a reversal. So you can see that individual has turned back to a dark morph butterfly now that the heat wave is over. And this is a very dramatic example. Of course, it doesn't happen quite as dramatically as this, but it gets the point across that A, it doesn't require genetic mutation to change gene expression, and B, it's quite reversible. And so there we have our two big guys, genetic changes and phenotypic plasticity. For the remainder of the talk, I'll just be talking about phenotypic plasticity, so keep your eye on the purple. And so how does phenotypic plasticity actually play a role in this climate change response? So there's a couple different ways that it can play a role. One, we can see what I've been showing, going from a suboptimal phenotype to the now favored phenotype in climate change. And that's great. That is positive. That individual will be more fit in its new environment and instant adaptation. So this is kind of cool because this might mean that plasticity has induced these adaptive environment or phenotypes, and no genetic change is actually necessary. Alternatively, we could see the second example here. So we can see that plasticity has induced a new phenotype. But what's interesting is that it didn't go to the optimal phenotype, but it did get lighter. So it's moving in the right direction, but it didn't quite get us there. So we might need a little bit of genetic changes to help that butterfly get to the optimal phenotype. And alternatively, phenotypic plasticity, it doesn't always work perfectly or the way that organisms would hope, and it can actually be quite maladaptive. So you'll see that this individual, when exposed to heat, actually went in the wrong direction. It got darker. And when phenotypic plasticity induces maladaptive phenotypes, this might actually increase the selection strength against genes associated with plasticity, ultimately leading to either like genetic divergence among these populations or even a loss of plasticity. And so for the rest of the talk, the way I'm going to kind of frame it is first, we're going to look at some of the consequences of phenotypic plasticity. So this will be my reproductive traits and did they change in response to heat? And then spoiler alert, it does. And so then we started looking at the mechanisms behind it. And I've already kind of hinted at the mechanism we're going to focus on today is DNA methylation. And then at the very end, we'll kind of talk about some of like the gigantic consequences. So does this phenotypic plasticity allow for instant adaptation? Is further genetic evolution required, or possibly even genetic accommodation occurring? And so we chose to look at that all in the umbrella of reproductive traits. And this is because previous research has really shown that elevated temperature can impact reproduction. So particularly take this example of these beetles here. So temperature stress early on in their life stage, so decrease fertility by altering gamete morphology. So heat was really impactful there. Alternatively, another example is take these butterflies here, and when you expose them to elevated temperature, what happens is that there's an impact on who they choose as mates, and then also when and how they lay their eggs. Both of them are not great responses, so both of them really are showing that elevated temperature negatively impacts reproduction in insects. And because reproductive traits just play such a key role in determining a species' response to climate change and really plays into some of the selection that are experienced in changing environments, we chose reproductive traits to focus on. And so first, like I said, we're going to focus on one of the consequences. So how does heat across the lifespan affect reproduction? And then the part two and the ongoing works of my talk will be looking at the mechanisms behind the consequences that we see in my first part of my talk. So let's jump in. Does heat induce changes in reproductive traits? Well, first, let's give you a little bit of background on life history traits of butterflies and moths. So butterflies and moths, as most of you probably know, because we oftentimes raise monarchs as young students, have very distinct life stages. So the egg looks very different than the larva, and then the larva look very different from pupa, and pupa look very different from adults. And because they have such distinct life stages, response to temperature may be oftentimes very life stage dependent. So for example, take the pupal stage. This stage is relatively immobile, so it can't crawl like the larval stage and it can't fly like the adult stage. So it's kind of stuck. So it can't behaviorally regulate heat. Additionally, what's really sensitive or possibly really sensitive to heat during the pupal stage is its development. Obviously, when butterflies and moths are in their pupal stage, they're undergoing metamorphosis. And metamorphosis has been shown to be particularly sensitive to heat. So heat at the pupal stage may impact reproductive traits more so than, say, heat at the egg or the larval stage. Additionally, temperature effects can be additive. So what do I mean by that? We'll take this example here. That means that temperature increase at multiple life stages. So in this example, heat across both the larval and the pupal stage results in these cumulative effects. So it's more detrimental to be exposed to two life stages than it is either one. And sometimes it's even worse than the just combination of the larval and heat stage. There's like some added negative effect of having it across multiple life stages. And so now that we've got that, let's look at my actual study system, the European corn borer moth. So why did we choose the European corn borer? Well, they are a widespread generalist herbivore and a frequent agricultural pest. So as their name suggests, they're from Europe. So they're an invasive species here in North America. And then they bore into our corn. So you can see here a larva inside a corn crop and also a pupa inside the corn crops. And because they are so disruptive to humans' agricultural requests, we know a lot about them. We've studied them to try and get rid of them. And in our process of studying them, we know their reproductive biology really quite well. And what's really important, particularly for the second and third part of my talks, is that they have substantial genomic resources, including a reference genome. And so the two kind of subheadings of questions that I'll be asking here is, does heat increase or decrease reproductive output? So is it positive or negatively affected by heat? And then are those effects additive at any temperature or life stage? And so to answer those two questions, we exposed moths to a bunch of different temperature regimes. So you can see at the very top here, we have the control moths. So they were never exposed to any heat in their life. So they were exposed to 23 degrees their whole life. But you can see the second one, they were exposed to heat for their entirety of their lives. And I just want to take a second here to kind of explain our thinking of 23 degrees and 28 degrees. So 23 degrees is our ambient or control temperature for the remainder of the talk. That is because that is about the average temperature that the average corn borer experiences in summer right now in, 2020, when that data was collected. Whereas 28 degrees, it doesn't seem like a big jump, right? Only 5 degrees Celsius. But in the grand scheme of things, that 5-degree increase is predicted to occur by the end of the century. So by 2100, the global temperature should increase by 5 degrees Celsius. And so that's the reasoning for our 23 being ambient and our 28 being our heated treatment. And that will be consistent throughout the entirety of my talk. It's always those two temperatures. And then the other one I want to just draw your attention to here is the bottom row. That is our additive treatment. So you can see that those particular individuals were exposed to heat during both their pupil and their adult life stage. So we were able to see if the combination of those two had any effect. And once we exposed them to their proper temperature regime, we put one male and one female in a cup, as you can see on the screen, and we let them freely mate for a couple of days. And each day we would come in and we would check the number of egg clusters laid. And that is a sign of whether or not they're mating, because females that have not mated with males in this species only produce like very single eggs, not clusters. So the indication of a cluster means that they mated. And the more that they mate, the more egg clusters they typically lay. And so, shocker, we, because I've focused on additive effects, you knew I had to find one. And so I did. We found that reproductive output was only impacted when we exposed them to elevated pupil and adult temperature. So what does that actually look like visually? So our control individuals, those that experience no heat throughout their entire lives, laid about 6.5 egg clusters after day 2. However, if we exposed individuals to elevated pupil and adult temperatures, this significantly reduced the reproductive output. It almost halved it. So that's a big difference. And so, yes, heat does decrease reproductive output, of course, only in some contexts. So it does require, as the second question answers, an additive effect. It does require both pupil and adult temperatures to be raised. And so then why might that be? Why does it take an additive effect? Well, take this horizontal lifespan of the corn borer, and I'll just throw up here some important biological processes. So at the beginning of their larval stage, they're hatching out of their eggs. I've already told you that during their pupation, they are undergoing metamorphosis the entire time. And then in particular in corn borers, male gametogenesis or spermatogenesis, when they're making their sperm, starts in the late larval stages, so when they're big fat caterpillars, and it goes just before they emerge as adults. But females are offset. So female oogenesis just begins at the very end of the pupal stage, and the majority of their egg maturation actually occurs after they eclose as adults. So, and then finally, reproductive behaviors obviously occur in adulthood when they're actually finding mates, choosing mates, and deciding where and when to lay eggs. So when we expose them only to the pupal life stage to heat, you can see that perhaps we only disrupted spermatogenesis. And a very similar story occurs when we only expose them to heat as an adult. Maybe we missed all of the male spermatogenesis and only disrupted oogenesis. But if you think about the combined effects, right, of pupal and adult temperature, then you start seeing that both gametogenosis were exposed to elevated temperature for the majority of those processes. But it's important to note here that we didn't, males and females were exposed to the same temperature treatment. So we can't parse out really, was it males or was it females? And we didn't measure eggs or sperm count. So this is more just a hypothesis as to why additive temperature had an effect. And then we see an effect now. So now we need to understand, or the pitch that we made was to understand the molecular mechanisms that are actually underlying this heat-induced changes in reproduction. And because I work with a lab strain that are pretty genetically similar across generations and across individuals, we kind of knew that it wasn't genetic change that's driving these differences between temperature treatments. So we thought it had to be some level of phenotypic plasticity and the molecular mechanisms that drive phenotypic plasticity. So let's dive into that with part 2 of what are the mechanisms that underlie this phenotypic plasticity. Alrighty. So DNA methylation in Lepidoptera, butterflies, and moths are incredibly understudied. Whole genome sequencing has only occurred in a handful of species. So for example, here's 2 of them, the painted lady and the Chinese oak silkworm. But you can see that whole genome sequencing has occurred in a wide variety of contexts. So the painted lady was sequenced in looking at the effects of larval crowding, whereas the Chinese oak silkworm was sequenced in the context of immune challenges. And because there's so few of them and they're all in different contexts, it's really hard to draw comparisons or look at commonalities among species within the order. What we can say, though, is from those that have done whole genome sequencing, what's really interesting is that methylation occurs within the genes. And if you work with vertebrates like humans, that's kind of in direct contrast with us because vertebrates tend to have methylation occur within promoters. But we're really seeing is that for invertebrates and insects in general, methylation occurs within the genes. So either in their exons or in their introns. For those, there were a couple that have looked at the effects of heat on whole genome or on DNA methylation. And you can see them here. So the domestic silkworm or the silk moth, they had plus 10 degrees Celsius, but they also looked at it in the context of humidity. And then we have the rice leaf folder. They had 12 degrees increase in Celsius. But what's important to note is that they exposed their moths to elevated temperature during the larval life stage. So we actually don't know what the impact of heat is on DNA methylation in other critical life stages, such as the pupal and adult life stage. And then there's also no studies that have examined the effects of heat and reproduction in the same one. So these 2 studies were looking at heat, but not in a reproductive context like I will be. And then finally, we don't really have a comparison between sexes, and this is within Lepidoptera. And this is quite important because in other insect species and orders, we find really great variation in DNA methylation patterns among the sexes. In some orders, we see that females have greater global DNA methylation. Some find it's greater in males, others find no difference. And we actually just don't know in Lepidoptera because no one has looked at it yet. And so the point of the second part of my talk was to try and fill in some of these gaps. So the broad theme is what are the mechanisms underlying heat-induced plasticity that we saw in the first part of my talk, but then also does DNA methylation change in response to a moderate heat? Because think back to what I just told you, everyone else has really done quite dramatic changes in heat, at least 10 degrees Celsius. We're going to try to do it in 5 degrees Celsius. Then we also want to see, do those responses differ among sexes? And if we do find changes in DNA methylation, are they in particular genetic pathways that we can kind of start to think about what pathways may be directly impacted by changes in DNA methylation? And so in order to answer these questions, we took DNA from the lower adult abdominal tissues. So we were targeting reproductive tissues in both males and females. Of course, we didn't just extract their reproductive tissues. So there were other tissues in there as well, but we really did try to target the reproductive tissues. And so we actually took 5 females and 7 males from the control temperature in part 1 of my talk. And then we also took 7 females and 7 males from the elevated pupil and adult temperature. So these were the exact moths that showed us reduced reproduction in the first part of our talk. And once we extracted that DNA, we used enzymatic methyl-seq to measure DNA methylation across the genome. So if you're not familiar with enzymatic methyl-seq, perhaps you've heard of bisulfate sequencing to measure DNA methylation. It's very, very similar. Instead of using the bisulfate conversion, enzymatic methyl-seq uses enzymes to convert it. It's just a little bit softer on the DNA, so you get better results. So that's why we chose to do this one. But it works in the same way as bisulfate sequencing. So how it works is take this hypothetical sequence here. You'll notice that some cytosines are methylated and some are not. And when you expose these DNA to an enzymatic treatment, what happens is the methylated cytosines remain methylated and remain cytosines, but the unmethylated cytosines turn into uracils, or what looks like a uracil, when you sequence them. And then when you sequence that, the enzymatic treated sequence, now that unmethylated cytosine looks like a thymine. And when you compare that to an untreated genome, or in our case, the reference genome, you're able to figure out exactly per cytosine, was it methylated and was it not? And then you're able to compare that between the 2 temperature treatments or between the 2 sexes to really get at a sense of where are differences in methylation across the entire genome. And so then once you get that sequencing data back, it undergoes a bioinformatic pipeline. So the first one, you trim and align the reads to our reference genome. Then you are calling the methylation state of each cytosine, and you're able to remove any SNPs so that doesn't cloud your data. And then finally, you filter low coverage, and then you do 2 big analyses. You first compare overall methylation patterns among the different temperature treatments and among the different sexes. And then you're also going to test for significantly differentiated or differentially methylated regions, and you're going to look at what genes those differentially methylated regions actually are located in. So let's first look at global methylation patterns. So on your screen now is the graph for females. And you'll notice that the x-axis is the temperature treatment, so either control or heat, and the y-axis is the percent methylation throughout the entire genome. One thing to note is that the percent methylation is really small. That was not shocking. That is pretty typical of insects. They just have very low global methylation percentages. But the striking thing here is that increased methylation occurred in these females after heat exposure. And the same thing occurred in males. So now you'll see the males have shown up as well, the same pattern. So heat exposure in males also increased global methylation. So our results indicate that increases in DNA methylation are not only observed in these extreme heats like I presented earlier, but also in response to a moderate temperature increase of 5 degrees Celsius, and may be more common than we previously thought. So that's now 3 studies that have shown increases in global methylation in response to heat. Additionally, what's really cool is that we did this obviously in the people and adult life stages. And so it allows future comparative work on life stage differences in methylation in response to heat. So perhaps all life stages in insect or in Lepidoptera when exposed to heat have increases in global methylation. Now I've switched it on you. So the x-axis, instead of being temperature exposure, is the sex of the individual. The y-axis stays the same at methylation percent. And you'll notice that the trends now look kind of different. Females, regardless of temperature treatment, always have higher genome-wide methylation compared to males. And this was really cool because, like I said, previous studies in other insects have found that females have higher methylation than males. So perhaps Lepidoptera fit in with this difference. However, because there's not too much to compare it to, we would really love to see other studies come out and look at methylation differences between males and females in other butterflies and moth species to really get a sense of if it's an order thing, so all butterflies and moths look like this, or if it's species specific. Our corn borers, unique in that females show higher methylation regardless of kind of temperature exposure or any environmental condition. Let's see. I already mentioned that. Yep, and further studies are just needed to confirm that. And so then the other question I told you we wanted to look at is what pathways show enrichment? So where are these differentially methylated cytosines occurring? And because we know from the previous part that these moths had changes in reproduction, we thought that these pathways may show enrichment. One pathway that may show enrichment are reproductions, and we were really interested to see if it was males or females. Could we parse out which of the sexes were driving the results in the first part of my talk? The second thing that we thought might come out was shifts would occur in functional pathways that have previously shown changes in gene expression. So not methylation, but gene expression after heat exposure. And a lot of cellular stress pathways come out, such as Hippo or Notch, if you're familiar with either of those stress pathways. And then finally, because we are exposing them to a change in temperature, we thought genes that play a role in responding to temperature change would be differentially methylated as well. And so first, let's just look at the differentially methylated regions. So this is the heat map from the males. And we can see that there is 173 significantly differentially methylated regions in males. As we predicted, the majority of them occurred within genes, so in exons or introns of genes, and the majority of them increased in methylation following heat exposure. And then I'll tell you, because it's not clear from the heat map that I show you, but 12 of those differentially methylated regions were in spermatogenesis genes, so genes that are known to play important roles in male reproduction in a wide variety of insects. So usually the silkworm or Drosophila, but they do have known roles in insect male reproduction. And females showed a very similar pattern, just less differentially methylated regions. So instead of 173, we got 73. The majority of these guys were also within genes, just like males. The majority of them increased in methylation, just like the males. And just like the males, 6 or a pretty significant portion of the 73 differentially methylated regions had known functions in oogenesis and female reproduction. So I would say check. We found reproductive genes like we thought, except for it occurred in both males and females. So once again, we weren't able to parse out if it was male fertility or female fertility that's driving the responses that we see in my first part of the talk, but perhaps it's both. So that's one more thing to explore as we move forward. We also found evidence for cellular stress pathways, so in particular the hippo signaling pathway, which is known to respond to different environmental stresses and regulate apoptosis in European corn borer, or not European corn borer moths, just in insects in general. So on your screen, you have key hippo genes. Those are just genes I can't build this pathway without. These did not show any changes in methylation in our guys, but the ones that just showed up in blue had decreased methylation in response to heat, and then all the ones that just showed up in red had increased methylation in response to heat. And this was kind of cool because hippo hadn't shown up in changes, hadn't really shown up in changes in gene expression or methylation following heat exposure in insects, but it does play an important role in metamorphosis. And our heat exposure occurred in metamorphosis. So perhaps we found hippo signaling come out as differentially methylated in our study because we're looking at the intersection of heat response during metamorphosis, which was kind of cool. So next studies that examine heat exposure, we really need another one that occurs during that pupal stage. So check. And then interestingly enough, we just didn't find much evidence for temperature response genes. And this may be because we chose just such a moderate temperature exposure. Typically like heat shock proteins and those guys, they really need a dramatic change in temperature, really, as the name suggests, heat shock. And we didn't really heat shock our individuals. So perhaps that's why temperature exposure, temperature response genes didn't come out as differentially methylated. So just to summarize, DNA methylation changes and it increases with heat. It's very different among sexes. Females, regardless of temperature treatment, have greater methylation than males. And also we saw different amounts of genes and different genes come out in response to heat. So the sexes are responding very differently to heat in European corn borers. And yes, we found some targeted genetic pathways, and the one that we thought was particularly cool and would like to see more research into was the hippo signaling pathway. And with that, that takes me to just the ongoing research that we have going for in my dissertation. And so this really is posed to answer the question, how does DNA methylation alter gene expression in insects? And that is because we have some previous literature on that. We found, but it's all over the place. So some studies have found that more DNA methylation results in increased gene expression. Others have found that the increase in gene expression is then dependent on where the methylation occurs, so whether it's at the 5' or 3' end of the gene. Others have found that more methylation results in stabilized gene expression. And then, of course, some found no correlation at all. So it seems highly variable in insects. And so we just need more studies to really kind of parse out some of these these questions, and maybe it is just species specific, but we need more studies in order to actually show that. And like I said, we're just really understudied in gene expression and DNA methylation together in insects. And so to help answer that question, we're looking at it in European corn borers. So we're going to look at it during the pupal stage. So we're going to expose pupa to heat or control temperatures. Then we're going to measure their transcriptome, so sequence their transcriptome. And the big key that differs from our second chapter or second portion of the talk is that we are also going to look at methylation and RNA data all in the same study. So we should be able to look at the correlations between DNA methylation and gene expression in the European corn borer. I can say that prediction-wise, the Asian corn borer just had some data come out in relationship to gene expression and DNA methylation. And they found a weak correlation between gene expression and methylation. So perhaps that's the best predictor of what our corn borers will do, our European corn borers will do as well. But it's up in the air. So this will actually be the addition to my dissertation that will be funded by the grant that I got from SSE. So it's really cool to see this addition come because of the funding that I was able to get from SSE. And so just to wrap it up, I've really talked about some of the consequences of the phenotypic plasticity and climate change. So I was able to observe fertility declines, but there's so many more things that we need to look at in terms of consequences. One of the big ones is transgenerational effects. I get that question almost every time I present my results from the first part of this talk. No, we did not look into it, but we do know that DNA methylation can be heritable in insects, so it'd be a really cool thing to study. And it's definitely possible that we would see some really interesting transgenerational effects if we did look into it. And that's on our radar of things to do next. And then also as we start to build our number of species and populations, what are the variations? Does everyone see fertility declines? Is there more nuance in it? What can we generalize? And we can see that same kind of parallel in the mechanism. So we do know that DNA methylation responds to heat. We've shown that it differs between sexes, at least in the European corn borer. But are these responses common? It's too early to tell. We just need more data and more species to really undergo this in order to start answering the ideas of common response. And hopefully I've convinced you that by understanding these consequences and the mechanisms, we can understand how phenotypic plasticity plays a role in climate change and what it might mean for species that are experiencing increased temperatures due to climate change, especially given that it's predicted that climate change is not going anywhere anytime soon. And with that, I'd just like to end with some acknowledgement. So obviously SSE and then the graduate student body that puts on these talks, thank you so much for inviting me today. And then everyone else that has made this possible. So obviously my lab and my PhD advisor, Dr. Kozak, the Dartmouth lab, because they have helped us with our colonies, GeneWiz, who helped sequence our DNA methylation in the second part of my talk, the Unity cluster, so our supercomputer that we can do our bioinformatics on, and then all the great funding that allows us to do our research. And with that, I will take any questions.
Thank you so much, Brittany. That was fantastic.
Let's see. I will shut my screen so I can see everyone.
Good idea. And we can always go back to that.
Yes.
I have a question, but I want to open it up first. We'll save my lots of claps and
yay.
Does anyone else want to go first with their question? If not, I will dive in. All right, I'll go ahead and ask the first one. It's a very quick. My first big question was about the relationship between methylation and RNA level, which you addressed before finishing, and I think that'll be really interesting. It's such a puzzle whether that does what we think it does or when it does what it might do. But going back to the very beginning of your talk, when you measured the fecundity, the reproductive output with the egg cluster numbers, is it fair to just measure number of egg clusters, or is there enough variation in the size of the cluster?
So I gloss over that for sake of time, but yes, we have looked at egg cluster size, and that is a really good use of overall fecundity. And we've studied it in different contexts, and egg cluster size does not change with temperature. It doesn't change when we give them like supplemental resources or anything. So we did do the groundwork on that. And yes, it is a fair use. But yeah, that's always a good question because if it varied, then we were skewing it.
Yeah. It looks like we have another question coming in the chat. I just want to say also that was such a beautiful explanation of the motivation for your work, the work itself, and your visuals are
Thank
just
you.
beautiful. Nice job on that.
Um,
Yeah, I had a question for you.
yeah,
It was a good talk. Thanks.
thank you.
I was wondering, have you looked at the viability of the eggs? We've done some temperature work where it seemed like the viability might be important as well.
Yeah, so we don't actually let them hatch out those eggs, but what I can say is that European corn borer moths, about 24 hours before they're going to hatch, produce a dark pigment that we call blackheads, and that indicates that they are going to hatch. And the percentage of like blackheads per egg clusters doesn't vary between the temperature treatments. So we would expect that at least percent viable eggs is about the same among the temperature treatments. But yeah, that's one of the things that if I could go back in time, I get that one a lot on my first part. So I would measure, are they viable? And then that gets into the ideas of like transgenerational effects as well.
I just wanted to ask, so I saw the beetle it has a wide distribution all the way southern Canada to southern United States. So they're receiving different temperatures now, maybe similar to the high temperature. So I just wanted to comment on that if you had any thoughts about those different temperatures in the natural populations, but then also uh, the in the egg part of the study, the moths that were exposed to something analogous to climate change would be the 25, 25, 25 moths, right? Because the climate won't uh, increase in temperature when the
Oh,
moth,
only in one life stage.
right,
Yeah.
right, the climate of the Earth so do you think that climate change will then not affect the egg clusters? That's sort of what that result was saying to me.
Yeah, so we liken to think, and I think I did a little bit better when I wrote that part, is that we likened it more to a heat wave. So heat waves will occur only at one life stage. So we kind of pitch it more as a life stage or a heat wave effect rather than like a total climate change effect. But yeah, that's one of the puzzling things of like when we exposed them to heat throughout their entire lifespan, they were okay. We didn't see that impact of elevated temperature. And so perhaps it's a level of like early accommodation or adaptation that kind of occurs in that early life stage that primes them to be okay throughout their entire life. Um, that's one hypothesis that we had. But yeah, it's very interesting that those guys that experienced the elevated temperature throughout their whole lifespan didn't have that reduction. We definitely thought they would. And it's up in the air and kind of still something that we think about in terms of what that might mean for corn borers.
It
You
just
could
made
imagine a scenario where climate change affected different microhabitats
Mm.
different extent too. So like if the place where the eggs are, the place where the pupae are, and the place where the adults are differentially impacted by the climate change, your segmentation of the exposure into life stages could potentially capture that. The fact that ponds might warm more quickly, for example, than the underside of leaves or something like that. I'm spitballing here, but um, that's what I thought of when because I had the same question that you're posing, Robert. At first, I was like, oh, why? How would the temperature change for part of their life? And then I was like, oh, maybe it's like that the natural history of where they're spending their time is differently, that the microhabitat is differently impacted.
Yeah, and that also plays into like we know that like springs and falls are warming faster than other ones, and they're only in certain life stages at different times of the year as well. So um and they undergo diapause as like late stage caterpillars. So that one might be a little bit more hardy compared to the other life stages as well, just given their life history.
There's a question in the chat now. Do you know how many, if any, of the methylation cases you discovered are reversible? And if you don't know,
Ooh,
do you have an
I
expectation?
don't. No, that would be so cool is to be able to measure them at heat and then take away the heat and see if anything goes back. That is such a good question. No, I don't know because we didn't look at that. Ooh, do I expect anything to go back? I would say yes. So our elevated temperature exposure for the second part of my talk was only about 13 days long to induce all those changes. So I would say that it's probably quite and able to be manipulated. So I would guess that if we exposed them and then turned off the heat, we could probably see quite a few of those differentially methylated regions go back to the original state. But that is such a cool question. I would love to explore that.
Well, I'm seeing people start to maybe pack up to go to their 10 o'clock or 11 o'clock or 12 o'clock or whatever o'clock it is, wherever you are, their next meeting. Maybe we can give Brittany one more round of applause and thank her for sharing this work with us today.
Thank you.
I really learned a lot. Thank you so much. And please tell folks that you know who might not have been able to be here that there will be a recording available that they can catch later. And yeah, I think, is there anything else? I forgot to mention the code of ethics in the beginning,
Oh,
Robert. Is
that's
there anything else
I
I'm
posted
forgetting
it.
to
Yeah.
mention
I think
now?
everybody saw it. So I think we're good. Everybody was very well behaved. So
Okay, yes,
appreciate it.
bravo.
Yeah. Good job, everyone.
Thanks on behalf of the audience. Thanks so much, Brittany, for taking the time to talk with us today.
Oh, yes,
Yeah.
thank you.
See you guys next week then. Thanks so much, Brittany. Really
Thank you.
appreciate it. Thank you, Sarah, for hosting.
My
See you guys.
pleasure.
Of course.
Bye.